We want to increase the awareness about the research within the rapidly growing field of translational medicine while highlighting the variety of disciplines within the field itself. Now, in the interest of time, as our first presentation will be a bit short and we're going a little off schedule, I will forego the introduction and hand it off to our first speaker, Dr. Andrew McCulloch, Director of the Institute for Engineering and Medicine.
Thank you very much, Rashan, and I apologize that I can't stay for this whole session because I have to introduce today's seminar speaker, Dr. Ackerman from Boston. But it's a great pleasure to kick off this session. And actually, you'll have a chance to hear more detail from Dr. Kaufman, so everyone will be a winner here. What I wanted to do was tell you a little bit today about the Institute for Engineering and Medicine, and what it's been doing, and particularly what it's going to be doing in the future. And Dr. Xu Chen was the founder of the IEM in 2008. He, of course, had a remarkable career and was voted onto all 4 national academies. And so I have the sort of daunting responsibility of taking over from him. But fortunately, he has made that particularly easy for me. So the IEM has 14 centers within the Institute, and 6 of them are disease and organ system oriented. 7 of them are technology focused, and 1 of them is the Whitaker Center for Biomedical Engineering that's directed by Dr. Watson, who has, of course, been very engaged in helping with events like Translational Medicine Day. And the Whitaker Center for Biomedical Engineering is the arm of the IEM that connects the researchers and the students with the industry in the San Diego area. We have 220 faculty members from 21 departments, and our mission is to accelerate the discovery of novel science and technologies to enhance healthcare through teamwork at the interface of engineering and medicine, and to advance the translation of innovative technologies into clinical application and commercialization. So what I wanted to do very, very quickly is just take you through just a little quick flavor of the type of work that's being done in the centers, and then finish with a couple of new interesting initiatives that might interest you. So I actually started the Cardiac Biomedical Science and Engineering Center that's now co-directed by Professor Evans and Ross. And this center really builds on our long history at UC San Diego of collaboration at the interface of cardiovascular medicine, physiology, and bioengineering. And so some of the technologies that we bring to bear to study heart diseases in CBSEC include imaging, tissue engineering, stem cell engineering, personalized medicine, computational modeling, and the development of new biomaterial-based therapeutics. And this is really one of our longest standing and most successful connections between engineering and medicine at UC San Diego, dating back to the 60s. The Musculoskeletal Research Center is directed by Dr. Sa and Professor Sam Ward, who's the Vice Chair of Orthopedic Surgery. And this center is really focused on biomechanics, instrumentation, imaging, and tissue engineering for conditions such as arthritis and muscular dystrophy. But also increasingly, we're interested in the CMSR in human performance. So not only studying injury and disease and recovery from injury, but also how elite trained athletes achieve their performance, and how we can help them prevent becoming injured in the first place. The Center for Advanced Neurological Engineering is directed by Professor Mobley and T.P. Jung. And this is really focused on engineering technologies for neurological applications, such as this portable head-mounted display for objectively assessing visual field loss. We have a Center for Engineering and Cancer, directed by Professor Howell and Professor Langfang Zhang in nanoengineering. And this is particularly focused on the development of nanomaterials and nanoscale technologies for early detection, diagnosis, and targeted treatment of cancer. Then a related center is directed by Professor Ada Al-Mutairi on nanomedicine and engineering, and it's particularly interested in biomaterials, so nanoscale biomaterials that can be used as diagnostics, as scaffolds for tissue engineered therapeutics, and also as contrast agents for imaging. The Center for Engineering and Diabetes, directed by Professor Goff and Professor Micah Sander in pediatrics and cellular molecular medicine. And it brings together engineering expertise in sensors and wearables and data analysis, particularly the long-term continuous glucose monitor developed by Dr. Goff's lab, but also regenerative medicine technologies, such as new tissue engineering strategies for using human inducible pluripotent stem cell derived beta cells and encapsulating them for the treatment of type 1 diabetes. I've been working a lot with the directors lately of the Center for Mobile Health Systems and Applications, because it's got sort of curiously a big boost from the pandemic. Almost overnight, the number of remote visits at UCSD shot up and exceeded the number of in-person visits for a number of months. And suddenly the need for remote monitoring and better access to wearables data really got a big boost. And so we've been trying to learn from the pandemic, and the Center for Mobile Systems and Health Applications has been promoting and the development of a number of new technologies to help address the pandemic and other health needs, which I'll talk about a little bit. The Center for Medical Device Technology is directed by Professors Tolke, Friend, and Barleben, and they design a wide range of medical devices, particularly for the cardiovascular system, such as stents and new algorithms and technologies for detecting thrombus from ultrasound imaging, for example. Surgical robots is another field. And I'll talk a little bit more about how the CMDT has also been working in response to COVID. The Biophotonics Technology Center is directed by Professor Burns and Professor Hall in radiology. Professor Burns was formerly director of the Beckman Laser Center at UC Irvine. And so he has long years of experience, but we also, our newest faculty member in bioengineering, Professor Lingyan Shi, is a biophotonics expert. She develops technologies for using stimulated Raman spectroscopy to be able to look at metabolic activity in living tissues, deep into living tissues. So this has huge potential, particularly in cancer therapeutics. Dr. Coleman and Dr. Sandy Ramos in obstetrics and gynecology direct the Center for Perinatal Health. And their focus has been particularly the development of novel technologies for perinatal and immediate postnatal monitoring in the pediatric intensive care unit, or neonatal intensive care unit in the NICU. Dr. Norbash, who's the chair of radiology, and Professor McVeigh in bioengineering, direct the Center for Cardiovascular Biomedical Imaging. They are developing a whole lot of new imaging technologies for the heart and vasculature, including high-speed, low-dose CT angiography, 4D flow MRI, and various CT and MRI modalities for getting more information with either less radiation dose or shorter MRI procedures. The Biomaterials and Tissue Engineering Center is directed by Professor Xiaochen Chen and Professor Louise Laurent. And its big thing is 3D bioprinting. And so this center has developed technologies for 3D bioprinting of liver, heart, vessels, tumor models, particularly as drug discovery platforms. We have a vaccine engineering center that's in the process of being rebranded into a broader infectious disease center in response to the pandemic. And it's directed by Professor Lay and Professor Victor Nizet in pediatrics and the School of Pharmacy. And they're particularly interested in, or have been particularly interested in novel vaccine developments, including vaccines for non-infectious diseases like atherosclerosis. But increasingly, over the last year, the scope of the center has, as you can imagine, with the emphasis on infectious disease, grown a lot. And then finally, as I mentioned, we have the Whitaker Center for Biomedical Engineering, directed by Professor Watson. Translational Medicine Day is in no part due to it's no small part due to his contributions in connecting local industry with our students. And as you organizers of this event know, our bioengineering students have been very active, not only in organizing Translational Medicine Day, BE Day, also Breakfast with Industry, and the San Diego and National Science Festivals are all major activities that our students play a leading role in. So we really appreciate the participation of students and value the resources from industry that Dr. Watson brings to help support these. Finally, I just wanted to show you about a few COVID-related projects that IEM has sponsored in the past few months or past year. They include the development of a new personalized medicine platform for frontline clinicians, particularly to give early warnings of burnout for healthcare providers, particularly in the early stages of the pandemic, but then again in the winter. Dr. Coleman and Owens have developed a wearable sensor for COVID-19 vitals tracking so that it requires less physical contact with patients and can be remotely monitored. Dr. Chrisman has developed immunomodulatory extracellular matrix hydrogels that are targeting the vasculature to reduce inflammation in COVID patients. We have Dr. Reek and colleagues from computer science and pediatrics and the hospital working on a low-cost mobile telehealth system so that patients and doctors can talk to and interact with patients without being in the room. And a medical electronic command center that particularly Dr. Kastner and Dr. Ramamurthy have been developing to have more efficient triaging and management in crisis situations, particularly at the nurses' station. And finally, Dr. Friend and Dr. Morris have developed this vacuum isolated locker that actually has saved 25 people in San Diego from having to go on mechanical ventilation. They all survived because by being in this locker, they were able to undergo high flow oxygen therapy without having to be intubated and without the risk of aerosolizing virus to the room. So that's just a little bit about IEM and our research activities. We also have student activities. Our Gemstones program stands for Galvanizing Engineering and Medicine Student Teams on Needed eHealth Solutions. That's directed by Professor Depp. What we're doing is finding community partners, particularly community groups representing underserved communities, and finding engineering solutions working on engineering solutions that UCSD engineering students can develop to help these communities. And finally, I want to mention at the graduate student level, UCSD is part of the Siebel Scholars Program. And in 2021, we're fortunate to have 5 engineering pre-doctoral students shown here receive Siebel scholarships this year. So with that, I want to thank you all for your time and energy, and the organizers for organizing this great session. I apologize that I can't stay and answer questions now, but I'll come back to the afternoon session and we'll be able to talk informally. Thank you all very much.
Thank you so much, Dr. McCulloch, for your very interesting presentation. So since we don't have any questions, I think we can move on to the next presentation, Dr. Kaufman. So to give a brief background for Dr. Kaufman, Dr. Kaufman is a professor in the Department of Medicine, Division of Regenerative Medicine, and the director of the cell therapy program at UCSD. Dr. Kaufman does clinical work in hematology and bone marrow transplant. His research focuses on the use of human pluripotent stem cells to study the development of hematopoietic stem or progenitor cells, lymphocytes, and other mesodermal lineages. In particular, his studies have developed efficient means to produce natural killer cells from human ES cells and iPSCs, suitable for new clinical applications to treat relapsed or refractory cancers, both hematologic malignancies and solid tumors. Current studies aim to translate use of these cells into clinical therapies and to engineer these NK cells with receptors to improve the killing of cancer cells. The title of his presentation is Improved Cancer Therapy Using Engineered Human Pluripotent Stem Cells. So with that, I will hand it off to Dr. Kaufman.
Okay, thanks, Rashan. Let's see, so supposed to share my screen here. Let's see if I can find my talk. Where'd it go? There it is. All right, oops, let's go to this, and hopefully this kind of, good. All right, does this work for everybody? Hopefully can see me. So despite doing talks by Zoom for a year, I still haven't figured out the Zoom background thing, so you get to see my home office. So thanks though for the introduction. As Rashan mentioned, I'm going to talk a little bit about the work in my research group that's really been developing over the last really decade or so of being able to use human pluripotent stem cells to produce a targeted cell-based cancer therapy. And as I'll show a little bit at the end, this has actually been translated into clinical trials, which now started a couple of years ago. And I am glad that I actually worked the word engineering or engineered into my title. I'm not an engineer, but I can play one for now. So just in terms of disclosure, these are some companies that I consult for, or actually co-founded Shoreline Biosciences, which is just getting started, Shihan and BusyCell. So I know this is kind of a diverse background, and so I just wanted to make sure everybody was on the same wavelength about cancer treatment. So these are the typical ways that cancer is treated. That is either by surgery, if it's a more limited focus, by radiotherapy, where again, it can be a more focused treatment, but typically maybe to a deeper or more challenging part of the body, like the brain, where you don't necessarily want to do surgery. Or if it's a more diffuse type of cancer, you use chemotherapy because that can essentially cover the whole body. And so these are, again, sort of the classic ways of doing it. You can tell that the slide is kind of old and blurry, but you cut it out, you burn it out, or you poison it. Let's see, moving forward, it doesn't want to move, there we go. Just because this is California, I want to make sure you understand how cancer is not treated. So if you go to the internet, you see a lot of things like these alternative cancer treatments of using healing energy to treat cancer. Don't do this. Go to a regular provider as much as people want to avoid doctors like me. We're here when you need us, and don't look at these alternative treatments. But what I'm going to really focus on today is a newer field of so-called immunotherapy. And so again, the top here sort of mentions the traditional methods that I mentioned. But here the bottom talks about more immunotherapy. So that is using the immune system or immune agents that activate the immune system that also provide a very powerful way of killing and potentially curing certain types of tumors. And this can be done, here it says sort of quote unquote, unleash the patient's immune system. That is, we can give drugs to improve the patient's own immune cells. Or as I'll talk about, we can actually make cells that are immune cells and administrate them to patients to cure their malignancies. So even though this is thought to be a new field, this is actually a figure or timeline that actually shows that this field of immunotherapy actually dates back 130 years. When the first type of immune therapy was done by a fellow named Coley, who actually found that certain patients with cancer who developed infections, that their cancer would go away. And so those infections actually rev up the immune system. Of course, they had no idea what was going on. They didn't know what lymphocytes were or anything at the time. But then they actually found that they could administer certain infections, bacteria to patients, and treat some cancer that way. Now, this isn't advised because patients can also die from those infections, but there are versions of this still done today. So there's a drug or agent called BCG, which is a killed bacteria or bacillus that activates the immune system. This is actually used for vaccines, for TB, tuberculosis. It's still used in certain parts of the world. But for some malignancies, most notably bladder cancer, so this shows here in 1976, they started instilling this into the bladder, and it could rev up the patient's immune system and actually kill the bladder cancer. And so now for almost 50 years, it's been used for that type of treatment. There's subsequently in the 70s and 80s, discovery and development of so-called monoclonal antibodies. These are targeted antibodies just like your body normally makes, but they could make them specifically to target and kill cancer cells. There's now a couple dozen anti-cancer antibodies that are used regularly. There's other drugs that are so-called checkpoint inhibitors. These are drugs that block immune signals of your immune system to activate it, and in the last few years have become a very important part of cancer treatment. But then in the last decade now, we've really gotten to this stage where we can engineer cells and use those for cancer treatment. And that's what I'm really going to talk about. And the most notable, I don't know, unfortunately, I can't see the whole audience, so I can't have you raise your hand. If you've heard of these so-called CAR T cells, which is really in the cancer field, at least my field, in the last 5 to 10 years, really kind of the biggest exciting advance. So these are engineered patient T cells or immune cells that can be, as I'll show you, taken out of the body, engineered with a specific receptor to target tumor cells and given back to the patients. So those so-called chimeric antigen receptors or CAR T cells are now FDA approved. And so I can, it's not as simple as this, but I can, write a prescription, or we can prescribe these treatments. One's called Kymira, which is from Novartis, and another one called Yescarta from Kite. There's actually a couple more that are approved now as well. And so we now, treat quite a number of patients with this type of engineered cell-based therapy. Let's see. Oh, there we go. So the idea here, again, is that we can take these genetic vectors or these viral vectors that contain genes for this, what's called this chimeric antigen receptor, CAR, that contains this antibody domain that recognizes a specific tumor antigen. And it's linked to signaling molecules that activate the T cell. And we can use this again to infect the patient's T cells and now target these against, in this case, a tumor antigen called CD19. It's actually on all B cells, normal B cells and malignant B cells. But we can, kill off all these B cells. You can kind of live without your normal B cells for some period of time. But it kills off all the tumor cells and has been quite effective at treating certain types of cancer. So the process that this manufacturing, or it's really a cellular engineering, involves is they actually take out the patient's blood cells. They collect their white blood cells. They expand or activate the T cells, which again are these important immune cell populations. They then can use one of various methods, typically viral-based vectors, to engineer the T cells with this CAR, and they give it back to the patient. This whole manufacturing takes about 3 weeks, give or take. But again, these studies were done in the last decade, that it showed, again, very promising results for certain types of lymphoma, where here, 54% of patients got complete responses in these early trials. And, these are patients who had failed many other therapies. And so this is sort of an example of what this looks like, that this is a bone marrow biopsy, and normally it's supposed to be nice and homogeneous, kind of like this, but you have this big blob of cells, which are actually lymphoma cells, and you then give them these engineered CAR T cells, and over a period of time, this all goes away. And again, you can see here that on the CT scan, you have these big nodules, which I don't expect you to know, what's what, but these are the lungs. This is the mediastinum. This is your trachea. But you have these big lymph nodes here that aren't supposed to be there. And you can see several months later, they get much smaller. And this just shows the activity of these cells, that now going out 2 years, about 70% of patients who get this treatment are still alive 2 years later, which for patients, again, with very refractory lethal malignancies is really quite impressive. So this has been, again, a big advance over the last decade. This is a very nice review. If you have one paper to kind of read about CAR T cells, this review by Crystal Mackel is very good. It kind of gives the timeline here. There's now hundreds of clinical trials using different CAR T cells in addition to these FDA-approved versions, but there's still challenges, right? This isn't a perfect treatment. Part of the problem is, 70 patients might do well. Many patients still relapse after this treatment. And moving this beyond these B-cell malignancies, the CD19 targeting, has been very challenging to do this against solid tumors, breast cancer, ovarian cancer, lung cancer, and so on. Really hasn't had the same impressive results. And so there's biological challenges, and then there's also sort of non-biological challenges. This is a very expensive treatment. It costs $500,000 just to make those cells from a patient. It takes 3 weeks to make them. Not every patient has 3 weeks to wait. Their tumor might progress. And this is an individualized based therapy, because your T cells have to be matched to yourself. Otherwise, you get toxicities of things called graft versus host disease and so on. This is really done as a so-called autologous cell therapy. So everyone is actually getting something different. So if one patient responds and one patient doesn't, I don't know if that's because their tumor is different, their cells are different that we give them, or some other variable. So it's still challenging. And so really my goal has been to make this type of cellular immunotherapy against cancer as easy to use as a drug. That is, it's targeted, which we have with these CARs, but that this is more standardized, that everyone gets the same thing. So if you get an aspirin at the store, or I prescribe you with a medication for your heart, everybody gets the same pill, right? It's all the same. It's not different. And that this is so-called off the shelf, that it's made, ready, stored, and available for the patient when they need it. You don't have to collect these cells, wait 3 weeks, come back, and so on. And we want this ready to go, again, like other treatment, other therapies. So the way that really now for the last 20 years, my lab has worked is using human pluripotent stem cells. Originally, I got into this just interested in learning how to make blood, or how your body makes blood, or could we make blood cells in vitro or in the lab the same as your body does. And so this actually started when I was a postdoc. I worked with human embryonic stem cells. These were developed in the late 90s. Hate to think what that means in terms of your age, but it's kind of like yesterday for me that I started this. But these are cells derived from the early embryo that can make all the cells and tissues of the body. And again, I started working on this to better understand how to make blood cells. In the 2000s, there was this development of so-called induced pluripotent stem cells. This was a method where we could take essentially other types of cells, like a fibroblast, a skin cell, a blood cell, and so on. And whoops, we could give it these a handful of genes, and we could reprogram it into a pluripotent stem cell that essentially looks and acts just like an embryonic stem cell. Again, it can make all the cells and tissues of the body. We can use various methods to make blood cells, differentiate them in the lab. We spent a lot of time trying to make hematopoietic stem cells that could be used as an adjunct or replacement for hematopoietic cell transplant or bone marrow transplant. That's still very challenging. But what we have gotten very focused on now, again, in the last decade or so, is making these immune cells, in particular a population called natural killer cells, or NK cells. So NK cells are a normal part of your immune system. This is what they look like. They're so-called large granular lymphocytes. They have these big granules. And they're part of your immune system that normally kills tumor cells and virally infected cells. And they do this, unlike T cells that have to be, matched to a specific patient, that these work as so-called allogeneic cells. So my NK cells will kill your tumor cells and vice versa. They don't need to be matched to a specific patient. And I won't go into all the activating and inhibitory receptors that these cells express that regulate this process. So this becomes very attractive because this doesn't have to be patient matched. Now, there have been clinical trials for 10 or 15 years using NK cells from different donors, typically unrelated or related or unrelated donors. And these have been shown to be very effective at treating patients with refractory AML, acute myelogenous leukemia. It's a type of leukemia where about 30 to 50% of patients who get these NK cells from various donors get into remission, some of them very long term. The problem is that, again, when we get these cells from different donors, they're kind of one-off products. It's still kind of individualized, and it's been hard to expand the use of these cells in a really meaningful way. Importantly, unlike the CAR T cells, which I didn't mention, which, while successful, can be quite toxic, and people can actually die from some of these related toxicities called cytokine release syndrome or neurotoxicity, NK cells seem to be relatively safe. They don't cause these same type of inflammatory syndromes. So we have this method in my lab, I won't go into details, where we can take these embryonic stem cells or iPS cells, differentiate them through a series of steps, and make a very pure population of natural killer cells or NK cells. And we do a lot of flow cytometry. This is a machine that looks at proteins typically on the surface of the cell, and based on these markers, we can see that we get this very nice population of CD56 positive cells, which are typical NK cells. And so this, again, when we first started doing this, I said we knew that NK cells were good at killing leukemia cells. So this is a mouse model with human leukemia cells, where the leukemia cells in this case are just injected under the skin. And if we don't treat the mice, the tumor grows. That's what's shown here by this bioluminescent signaling. Kind of looks like a weather map. But if we give, in this case, we're using NK cells that we could also make from umbilical cord blood as kind of our control. We gave them just one IV injection of these cord blood-derived NK cells, and the tumors grew a bit less. But when we use these ES-derived NK cells, the tumors went away completely. 13 out of 13 mice in this original study, just using one injection of NK cells. So that got us very interested, again, at what we could do with these cells. We later tested this against more challenging models. I mentioned that solid tumors tend to be more challenging. This is an ovarian cancer model where, again, here they get the tumor cells intraperitoneally, kind of in the belly of the mouse. They get, again, one IP intraperitoneal injection of the NK cells, either now from peripheral blood or from iPS. And with the peripheral blood, they go away a bit. With the iPS, they go away even more. But this sort of shows the challenge now of trying to target and kill ovarian cancer, or these solid tumors, is that whereas we could essentially get rid of all the tumor cells if they're leukemia, we need to do something more with the ovarian cancer cells to kill them off completely. They're a bit more resistant. So it kind of gives us a platform to build on. And that's really what we've been working on now in the last number of years, that we can use different strategies. And because of time, I don't have time to go through all these, but I'll give you a couple examples. One is we can actually engineer these iPS-derived NK cells with CARs, with these same chimeric antigen receptors like are used in the clinical T cells. We can engineer other proteins which improve the function of the cells, or we can knock out specific genes that normally inhibit NK cell activity. So this is sort of the idea that, again, we can take these pluripotent stem cells, add genes for these CARs, differentiate them into CAR-expressing NK cells, and target the tumor cells. And again, we can get a very homogeneous population of cells. We can, characterize them all we want. It's not just a one-off product like we get with the CAR T cells, but again, we can make this in a very uniform manner. This just shows the characteristic of these CAR NK cells. They look like normal NK cells. And then, so we tested these in vitro, but then we tested them in that ovarian cancer mouse model. And this is sort of the model here. Again, we give the tumor cells intraperitoneally, then we inject the NK cells. If we, again, if we just have the tumor alone, the tumor grows. If we use, again, NK cells from peripheral blood, unmodified iPS cells, or I won't get into if we use a CAR that is made more for T cells, you do get some anti-tumor activity. You can, again, see that the signal gets less, but invariably the tumor comes back within a few weeks. If we use these now specific NK-specific CARs that we design, we can get much deeper remissions. You can see in these mice, some of the tumors go away completely, and much more prolonged remissions. And so this seems to be a much better strategy. And we can actually give several, we don't show here, we can give repeated doses of these cells and actually in some cases completely eliminate the tumor. And so this just shows with the one dose, again, with what I was showing previously, we were kind of up here. Now we're kind of down here. So we get much deeper remissions and significantly prolonged survival with just one dose. And again, other systems, we can actually cure some of these mice using this approach. It's just some other strategies. So the other method that we've used to improve the NK cell activity is, again, we not only can express new proteins on the surface of the cell, but we can knock out genes inside the cell using the so-called CRISPR-Cas system, which you might have heard about. It's become a relatively efficient way of engineering cell products. So we knocked out this gene called CIS or CISH, depending if it's the protein or the gene. This is normally an inhibitory protein. I won't get into all the details. It's downstream from IL-15, and it kind of turns off this activation signal. And it was known from mice that if you knocked out this gene, you got NK cells that had improved anti-tumor activity. So we did the same thing in our iPS-derived NK cells. We can just show here that we knock out the gene and protein completely. They function better. We get a whole bunch of different gene expression. I won't go into all the details, but just by knocking out, it is kind of always interesting to me. We can just delete one gene out of these cells. You have about 20,000 genes. If you just knock out one, you actually get a significant up or down regulation of over 3,000 genes. So it just shows how complicated the regulatory aspects of this is. Over 10% of your genome is affected just by changes in this one gene or protein. Then we took these cis knockout NK cells, and again, this is now using our leukemia model, where again, if we don't treat the mice, they die. If we give our unengineered NK cells, we get better survival. But with these cis knockout NK cells, again, we could get even better anti-tumor activity, and again, in some cases, even cure the mice from these leukemias. So this really, again, has a lot of advantages of using this iPS-derived NK cell system. We can make this standardized off-the-shelf system. We don't need to get cells from specific patients or donors. And, we think we can get a better cell therapy with less toxicity. And we can potentially add many of these engineering steps together. So I had previously collaborated with a company, they're actually here in town, called Fate Therapeutics. I'm not part of them now, but they've actually translated this type of technology into clinical therapies. And so they now have at least 3 clinical iPS-derived NK cells in clinical trials. One is called FT500. This was the first sort of unengineered iPS-derived NK cell that we actually treated the first patient here now 2 years ago. This is actually the first iPS-based therapy done in the US and the first iPS-derived blood cell or cancer therapy done anywhere in the world. So I was really thrilled that we can do that here at UCSD. They now have 2 versions of these engineered iPS NK cells, one with this protein I didn't talk about called CD16, the other with this NK cell specific CAR, and so on. They don't have the cis knockout. That's actually being done by my new company called Shoreline Biosciences. So these have all now actually started clinical trials. And this is actually the first patient. We do have permission from the patient. This is actually the first patient who was treated with the so-called FT500, the first iPS-derived NK cell. And this is actually the bag of cells. So I say that's about 20 years of work into that one bag of cells there. But glad that got moving forward. Whoops. So this is, I'll just kind of stop there. I did time this more for the half hour. I guess, with Dr. McCulloch stopping early, we got a little more time for questions, but we can move on a little earlier as well. But this is my lab prior to the last shutdown, but a somewhat allowed outdoor lunch or happy hour there wearing masks. But have a lot of important people who contributed. Ethan Lee, who did a lot of the iPS CARs, Huang Zhu, who did the cis knockout, a host of others, people who really pioneered these iPS NK cells are here in black, and collaborators both at the University of Minnesota, where I was before I came to San Diego, and at Fate Therapeutics. So I'll stop there for questions, or you can always contact me offline separately if people are interested in this.
Thank you so much, Dr. Kaufman, for your presentation and for agreeing to present ahead of schedule. So now, does anyone have any questions? Feel free to type it in the chat or unmute your mic.
I have a quick question. So, sorry. So I remember you saying that the CAR T cells or the CAR T therapy was really costly to produce. So how much less cost is there for producing the NK cells? And is it feasible?
Yes, so it is. So, again, Fate with these initial products, they did a manufacturing system where they could make several hundred doses, somewhere around 10 to the 11th, 10 to the 12th cells, all in one batch over a period of, I think, about, 6 or 7 weeks. And, again, this isn't I can just say what's kind of been reported. But as opposed to that, $500,000, their manufacturing cost per dose is probably more on the range of $5,000, right? It's not necessarily what it's going to cost the patient, but it's perhaps almost 2 orders of magnitude less by doing it at this type of scale, and that may only get better, right? So this really has been sort of priced out that it is going to be much more feasible to do it in that manner.
Okay, thank you.
Oh, somebody's asking here. Here we see. Many clinical trials tested in combination with other therapies. So typically they get this type of, depending on the tumor, they'll get it after chemotherapy. So these are often patients who have had chemotherapy before, and it is kind of combined with chemotherapy in that, I didn't get into the details, but because these are allogeneic cells, your body would otherwise reject them. We actually give some chemotherapy to kind of suppress that immune response and let the cells survive better. Not typically given with radiation, and I'm not sure what the CBIs are, checkpoint inhibitors, something like that. I think there are trials actually combining these with antibodies or checkpoint inhibitors, and so on. So those are kind of ongoing, but there is a lot of lab work that we can actually combine these with antibodies, and so on. That makes a lot of sense. All right, anything else? So good. So you guys are all undergraduates pretty much, is that right? I don't want to misspeak, but we're graduate students.
Yes, I believe we are all undergraduates.
Undergraduates, okay, well, good. If people are interested, this might be different than some of the other bioengineering, but always, you know, once you can, I don't even know if people are on campus now or not, but I've had a number of students in my lab, so hopefully this is interesting.
So are there any more questions?
Okay. All right. Well, if not, I'm probably going to get off now, and folks can always contact me separately.
Okay.
Okay.
So that will wrap up this speaker session. Next up, we have speaker sessions number 2 and industry demos from 2:50 to 3:50 p.m. Feel free to join whichever event interests you. Thank you so much, you guys, for coming.
Yeah, thanks for letting me join you. Thank you.